The last topic for today, this topic really important and interesting, medical genetic past, present and future. A personal view, Emeritus Professor Michael Patton from St. George University of London. Upon this time occasion, I would like to invite Emeritus Professor Dr. Wasun Chantratita, Head of Center for Medical Genomics, Faculty of Medicine, Rama Hospital, Mahidol University, and Professor Dr. Manop Pithukpakorn to be moderator for this topic. Please invite.
So let me introduce the Professor Michael Patton. He is graduated from University of Cambridge and Edinburgh and completed his clinical training in medical genetics at Great Ormond Street Hospital. He then moved to the St. George Hospital in London, where he set up the Regional Genetic Service and Academic Genetic Department. He has had a research interest in gene mapping and the malformation syndromes and worked extensively on Noonan syndrome. He has published over 200 papers and contributed the discovery of over 20 disease genes. He was Dean of Enterprise and Innovation at the university, and more recently was the Group Medical Director for HSL Pathology, which is a joint venture between University College Hospital, the Royal Free Hospital, and Sonic Healthcare, and was responsible for bringing genetic testing into mainstream pathology. So, ladies and gentlemen, please welcome Professor Michael Patton.
Good evening. It is great pleasure to be invited to speak at your conference in Bangkok this evening. I very much hope to be able to meet you in the future, but obviously things are difficult with the COVID crisis at the moment. Most medical specialties have developed over hundreds of years. For example, it's taken 400 years from the discovery of the circulation of the blood to the current state of knowledge in cardiology. But medical genetics has developed over one professional lifetime, about 40 to 50 years. And I have been involved in medical genetics over most of that time, and I'd like to show you how the speciality has developed using examples from my own career and to speculate where it may go in the future. I started my medical training at the University of Cambridge, but what I'd like to talk to you about is a personal story of how perhaps I became interested in genetics for the first time. When I was an undergraduate, I went out with this girl called Gabrielle, who was an art student who lived in Cambridge. And one day she asked me to join her with her parents for Sunday lunch. Now, as most young men in the audience know, this is often quite an intimidating experience, but it was particularly an intimidating experience for me, because it turned out that Gabrielle Crick's father was Francis Crick, a Nobel Prize winner and the discoverer of DNA with James Watson. It was clear that this was not going to be a small talk, because his first question to me was, what did they teach you about genetics in medical school in Cambridge? And I thought for a moment and answered that once they taught us about chromosomes, and I thought that would impress a Nobel Prize winner, but far from it. He began on a rant about how little doctors knew about genetics and said words that I still remember today, that genetics will be the most important part of medicine. Not just interesting or not just important, but the most important part of medicine. And that for a 19-year-old medical student was quite an insider tip, and certainly had an influence in my understanding of genetics from that moment forward. I should probably at this stage say that I did not go on to marry Gabrielle, and that my future career did not develop through nepotism or particular undue influence from Francis Crick. I went on after doing my preclinical in Cambridge to Edinburgh and became interested in pediatrics. And when I finished my house jobs, I decided to do a degree in genetics in Edinburgh. And I remember telling the surgeon I worked for that I was going to do a degree in genetics. And he said, "Well, Patton, I'm sorry you're going to leave medicine. I thought you might make quite a good doctor." The genetics course I did in Edinburgh was with the MRC, Human Genetics department. It was still very much early days in genetics, and very few doctors were involved in the field. But we did have good diagnosis for some of the genetic disorders by looking at the chromosomes, and the technique for looking at chromosomes in some ways has not changed very much from that time, in that the cells have to be cultured, dropped on a slide and stained to spread the chromosome karyotype out. But the rest of genetic diagnosis was very primitive. There were a few genes that had been mapped to particular blood groups, but there was no more than that. And DNA hybridization would be unrecognizable today, because DNA hybridization was looking at the melting point of DNA. When you mixed DNA from different animal species with human, you found that the closer the melting point was to human DNA, the closer the DNA sequence was thought to be. Certainly a long way from any diagnostic use. But I think the ability to diagnose Down syndrome as trisomy 21 and some of the other chromosome causes of mental handicap like Cri-du-Chat and Edwards syndrome were certainly a major step forward and made pediatrics and pediatricians interested in genetics. Having done a degree in genetics, I then went on to general training in general medicine and in pediatrics, and had the opportunity of coming back into genetics in 1981 to work with Professor Peter Harper on myotonic dystrophy. I carried out clinical studies on myotonic dystrophy with most of the families in South Wales, but the laboratory basis of the study was looking at red cell membranes, because we felt that the myotonia was probably related to some biochemical or structural change in the cell membrane. Unfortunately, that work came to nothing, but I had, by good fortune, also stored DNA from all the families, and it was that collection of DNA that helped later to identify the gene for myotonic dystrophy. Following this, I then went to Great Ormond Street for more formal training in medical genetics, and had the opportunity of working with Michael Baretza and Robin Winter on the London Dysmorphology database. And we went through the library on regular occasions, looking out all the references on syndromes. And indeed, in clinical practice, we were seeing many children with multiple malformations and describing new syndromes on a regular basis. It was certainly a great time in clinical dysmorphology. But there was also other things going on at the same time. It was the beginning of the human gene mapping project. And although a lot of the attention has been focused on the work by Craig Venter and Francis Collins, in reality, quite a bit of the human gene mapping was done by different international groups in collaboration, working together. And it was an exciting time because we were mapping genes at a regular rate, rather like the age of exploration when Christopher Columbus could go to America and put a flag on the map. We were finding genes and putting flags in the genomic map. The work moved faster than people thought, and the final first draft of the human genome was published in 2001. And it was celebrated by both Nature and Science with special editions of both magazines. Just at a personal note, this slide on the left shows the number of genes that were mapped when I started my work at Great Ormond Street. And by the time I'd finished, there were about 50 to 60 genes mapped. And I had contributed to the mapping of X-linked nephrogenic diabetes insipidus and myotonic dystrophy. But throughout my career, I've continued to work on gene mapping, and in collaboration with my colleagues in the department at St. George's, we have probably mapped over 25 genes in the human genome. And that is actually a bigger contribution than we might have thought, because originally it was taught that there were 100,000 genes in the human genome. And when the human genome was first published, it was found there was only about 40,000. That was much less than predicted. And when we looked at the genomes of other species, it turned out that the human species had only twice as many genes as the nematode worm. And to be honest, the nematode worm is a rather unexciting organism, and it certainly doesn't do astrophysics and write sonnets, but yet it has half the number of genes that we do. So what has gone wrong? Are we underestimating the nematode worm, that it's cleverer than we thought, or are we as a species more stupid than we had thought? The answer is not how many genes you've got, but how you use your genes. And I think the best analogy of this is to think about the 26 letters in the English alphabet. It hasn't stopped great authors like Shakespeare from writing sonnets and plays. He never complained that he hadn't got enough letters to do his job. The other thing that came from the human gene mapping is the understanding that we are perhaps closer to our primate ancestors than we had thought, and there is only 2% difference between ourselves and chimpanzees. And the question, what is that 2%, is a fascinating question. It is probably the genes for language development that have made the evolutionary step. The genes that allow us to express concepts and communicate ideas without physical objects has greatly enhanced our evolution. But really, we are still, even at the end of the human genome draft, we are still at the early stages of understanding the human genome. It is rather like, in terms of child development, understanding single words and not being able to put those words into sentences or to write in paragraphs. we still have a lot to learn about how genes interact, what controls gene expression, and ultimately how we can modulate gene expression in the treatment of genetic disease. After completing my training at Great Ormond Street, I was fortunate enough to be appointed at St George's Hospital in London. At that stage, there were a number of genetic centres, but many parts of the country had no medical genetic service. And my remit in being appointed was to set up a genetic service for a population of 3 million people living in southwest London, Surrey and West Sussex, and also to set up an academic department. But the problem was I was single-handed, and on my appointment I didn't even have a desk to start with. It was quite an uphill struggle, often very exciting, but we made it in the end. I was able to move a cytogenetic lab from one of the long-stay mental handicap hospitals into the hospital at St George's, and we started with some molecular testing in the academic department of pediatrics. The model that I had was one which I think many medical genetics centres have adopted. We had really 4 parts to developing the service. We had the clinical service, and I started doing outreach clinics and gradually increased the number of geneticists and genetic counsellors over the years. We also had the cytogenetic lab, which was on site and working well. The molecular lab started from scratch and was quite a challenge to keep up with the developments that were happening in molecular genetics. And we also set up a research department, and I think it's very valuable to have the research integrated with the clinical service, because the research questions you answer are more clinically relevant, and the opportunities for research questions come very often from the clinical material that you're seeing. Gradually the service did build up, and when I eventually retired, we had about 120 staff in the department. In the cytogenetic service, one of the early innovations was fluorescent in situ hybridization. And this came in for some of the microdeletion syndromes. And here we've got an example of Williams syndrome, which is due to a deletion of the elastin gene on chromosome 7. And you can see in one of the slides that the elastin gene probe is missing, but the control probe is there, which was a way of diagnosing the syndrome, which would not have otherwise been possible with routine chromosome karyotyping. But the fluorescent in situ hybridization technology advanced very rapidly, and very soon we were doing what is known as a comparative genomic hybridization, or a chromosome microarray. And this basically labels the patient's DNA with a red fluorescence and the reference DNA with a green fluorescence. And the reference DNA, which is taken from an average population, has about 40,000 drops of DNA from across the chromosome spread. If the two match equally, the red and green form an orange dot. But if there is a duplication of the patient's DNA, then that will come up with a red dot. And if there is a deletion of the patient's DNA, that will come up with a green dot. And this is what it looks like in practice. But fortunately, we do not have to actually read and score all these individual dots under the microscope, because that can be handled by a software program. Here we have an example of how the microarray is presented for analysis. the software has aligned all the dots of DNA along the main horizontal line, but in one point you see the dots have above the line, and this represents a duplication on chromosome 7. And the points are allocated along the gene map at the bottom. This would cause polydactyly and holoprosencephaly and would not be picked up by conventional chromosome analysis. And it's just one example of how useful the microarray data can be. With our molecular testing, it was quite a challenge to provide a service from a small department. We were very pleased right at the beginning when the gene for cystic fibrosis was published that within 6 months we had a service up and running testing for the common mutation delta F508. But after that, the number of genes that were available for diagnostic testing increased literally by the week, and it was difficult for us to keep up to date. We had at the end a number of the common disorders like cystic fibrosis, Fragile X, Huntington's disease, muscular dystrophy, and then we shared some of the rarer disorders between different labs, so that we did Prader-Willi and myotonic dystrophy and swapped that with other labs. But at the end of the day, the testing had increased. We'd gone from not just looking at single mutations or even single genes, but to looking at whole panels of genes, which can be very useful if you're looking, for example, at something like retinal dysplasia, because there are all sorts of different retinal diseases and very difficult to distinguish between them phenotypically. Although the testing has got more complex, the sequencing has got cheaper and certainly more rapid. And so it is possible now to offer more sophisticated testing. The one that I think is particularly useful clinically is the whole exome sequence. And this has been particularly useful for children with unidentified malformation syndromes. The problem about doing whole exome sequencing, where you look at the express sequence of all genes, is that you get a lot of variation, and some of it is not significant clinically. You can reduce this variation by looking at the parents at the same time in what's known as a trio. And any abnormality or variation found in the parents and in the child can be taken as a familial variation. And this may reduce the number of variations found to a small number, which may then be recognized through bioinformatic analysis as being pathogenic. But we still have a number which will be classified as variants of unknown significance, and this still limits the use of whole exome sequencing. A lot of people say, well, what about sequencing the whole genome, not just the expressed exons, but the whole exons and introns? The problem at the moment with this is that it creates a very much larger amount of data, which is difficult to analyze because the variation may not have been seen before. And the way to improve this is to test this against large population studies like the 100,000 Genome Project and others in different ethnic groups. And I think it will be a matter of time before we can use whole genome sequencing as an everyday tool in medical genetics, but it will certainly arrive as a gold standard in due course. I think some people put too much faith into whole genome sequencing. It sometimes has been said if you tested every baby at birth with a whole genome sequence, you'd know exactly what diseases they were going to suffer throughout their life. In point of fact, I think this is absolutely incorrect, because most of the diseases we see in later life are not directly due to a single gene, but are related to diet, degenerative changes, and in the case of many cancers, a random somatic mutation. We won't be able to predict these by whole genome sequencing, and we'll certainly need other approaches in investigating these diseases. One of the features I found running the regional genetic service was that there were a lot of changes had taken place since I first started. It was becoming increasingly difficult to run a comprehensive molecular testing laboratory, and with the competition from large commercial labs and large nationally funded labs, it is probably going to be the case that much of the testing will devolve to the larger laboratories. The other thing was that genetics as a subject was increasing in all directions, and it was difficult to be an expert in all areas of genetics. And in some areas, like cardiology and neurology, other specialties were becoming very much experts in genetics. A final change which made a difference was we were moving from diagnosis towards treatment, and most of us in medical genetics had been very much trained in the area of counseling and diagnosis rather than in direct medical treatment. So I think we've got to look at how the genetic centers will evolve, and there'll be different solutions in different parts of the world. In 2013, I retired from the NHS and had the opportunity to take up a very exciting post as Group Medical Director for a new venture, which was to build a 15-story laboratory in central London. It was a joint venture between Sonic Healthcare, which is one of the largest diagnostic companies in the world, together with two of the largest teaching hospitals in London, University College and the Royal Free. And the remit was to put all the pathology services together in this building to cover those teaching hospitals, 5 other NHS trusts and about 60 private hospitals. And the new build was in an excellent position. It was just opposite the British Library and the Crick Institute, and it had excellent facilities. These are the pictures of the building from the architect's point of view, and as you can see, the offices which I ended up in had this wonderful view over London, looking down over St Pancras Station. And we had 2 of the layers of the building with gardens in central London, but of course the main part of the building was these open plan laboratories on all floors of the building. But the challenge for me was to see how we could integrate genetics into mainstream pathology. We allocated 2 floors of the building to molecular pathology, and we integrated all the, I think it was 14 molecular labs in the teaching hospitals into one lab. And this meant we had human genetics, virology, hematology, parasitology, oncology, and other specialties all together. And the way we organized it was a functional lab which had mass DNA extraction, DNA sequencing, and then rooms for analysis and interpretation. We had a few separate labs for highly infectious samples or where we had to avoid any contamination. But one of the advantages of this is we shared expertise between different specialties and found we were all talking the same molecular language. And because some of the specialities were dealing with urgent referrals, we operated 24/7. The integration of the molecular service also allowed us to carry out translational research and develop new diagnostics. And one of the areas we were at the forefront was in cell-free DNA prenatal diagnosis. We worked with Ariosa in setting up the Harmony test, which allows some of the cells from the placenta, which cross into the maternal circulation, to be studied for the circulating DNA, and allows diagnosis of Downs and other chromosome aneuploidies from a maternal blood sample at 10 weeks. And this non-invasive prenatal testing will, I think, have a wider application in other areas of pathology in the future. I think the next point is to answer the question, where will medical genetics go from here? And obviously this is speculation, so these are my own personal thoughts. I think there are two ways of looking at this. One is to look at extending current trends, and the other is to look for new technologies or new paradigms which might change genetics in ways that are difficult to predict. One of the existing areas that will continue to expand is the use of gene therapy. The early trials with gene therapy led to a number of complications, but now with targeted gene therapy, we've been seeing some very successful treatments for X-linked immunodeficiency, forms of leukodystrophy and hemophilia, and I think we will see others in the future. And where we have a gene that is overexpressed with an abnormal protein, the use of RNA silencing treatment, as in Huntington's disease, is showing great promise. The other area of treatment which will show great promise is stem cell therapy, or as I think it should probably be better called, tissue engineering. The term stem cell therapy, when it was originally from fetal cells, caused a number of ethical and regulatory problems. But when we think about the majority of human disease, it is degenerative rather than due to single gene causes. And I think tissue engineering will play a major part in these degenerative disorders. It has already been used with some success in conditions like retinitis pigmentosa, and will be used in a number of genetic disorders. But the thing that has limited its use at the moment is the difficulty of developing stem cells, as most of it is being done in research labs. I think, however, with industrial investment and development, the costs will come down and the efficiency of the treatment will greatly improve, and we will see it being used widely in the future. We will also see advances in cancer diagnosis and therapy, and this example from a paper by Mike Stratton some years ago shows how exciting this can be. He studied a patient with malignant melanoma and found the patient had a BRAF mutation. And this is one of the genes in the RAS-MAPK pathway, and there was a commercial inhibitor for this, and when this was given to the patient, the metastases which you see on the left virtually melted away in a few weeks. The patient, unfortunately, was not cured long-term because the melanoma then went on to get another mutation in a different gene, and once this happened, it was out of the control with the drug inhibitor. But I think it shows the power of specific oncogene diagnosis. We'll also see this approach changing cellular pathology. Traditionally, histopathology has looked at H&E sections to try and identify the anatomical origin of cancer and its aggressiveness. But that will now be combined with genomic expression studies and identifying specific oncogenes in particular tumors, because it may be that it is not the anatomical site that's important, but the oncogene behind driving the cancer that's important when it comes to treatment. It will also come with advances in cell-free DNA diagnosis. When a tumor is removed, it will be possible to look at the oncogenes in that tumor regularly by doing blood samples every few months to see if recurrence has occurred, long before there's any lump or tumor found. And if an early recurrence is found, treatment with chemotherapeutic agents can be tailored directly to the mutation of the oncogene. And we may take this even further, rather than in treatment, in trying to prevent cancers by screening, by looking at oncogene panels in cell-free DNA diagnosis samples in the population. It is perhaps the holy grail to be able to replace breast screening, bowel screening, prostate screening, cervical screening by one regular blood test. And that day may come at some stage. But I think the paradigm shift in the future will come from the digital integration of clinical and genomic data. We are, I think, all familiar with the use of bioinformatic data in genomic analysis. Very often the phenotypic data is rather poor, but it should be remembered that our clinical data now is also digital. Electronic patient records, physiological variation, and imaging all has a binary basis and a digital form. And we can integrate the genomic and the clinical data together and start to look at the dynamic balance of genetic disease with the physiological and environmental variation. And this will allow us to move from studying single gene disorders into understanding multifactorial disorders in a dynamic way. I think all sorts of opportunities might open with this, and I've put here the possibility of looking at gene expression with imaging. On the left, we've got the technetium scan, which shows the possible increased inflammation in the ankle, suggesting either infection or tumor. But we could have a much subtler gene expression looking at the brain to see over time which genes switch on and switch off in psychiatric disease, which would greatly change our understanding of that area of medicine. And what about considering the combination of nanotechnology and gene therapy? Most of the disorders that we suffer from are disorders of disturbed homeostasis. And we're familiar with the cardiac pacemaker, which picks up cardiac arrhythmias and automatically corrects them by electrical activity. It is a form of autopilot, and maybe we could develop monitors which correct with gene therapy certain abnormal physiological states. Could we perhaps improve diabetic control by having monitoring of glucose and the gene expression of insulin matching minute by minute? Could we find a way of correcting obesity? Could we perhaps help the immune system detect and destroy cancer cells before any visible tumor develops? And could perhaps in the future we find ways of reversing the effects of aging? I think I'm probably going beyond the realm of science into the realm of science fiction, but I hope that some of these ideas have stimulated you this afternoon. I think what Francis Crick told me when I was an undergraduate has come true. Genetics is now the most important part of medicine, and I think there are few who would question that.
Sir, it is my great honor to have listened to you today, sir. It's surprisingly that you starting from cytology and extended to the exome, whole genome, cell-free DNA, NIPT based on harmony, cancer-targeted therapy, nanotechnology, and gene therapy. Also the digital integration that perhaps we need more computing power. The question from the floor is, what about wellness at the genomic level, such as the nutrigenomics, exercise, genetic testing, microbiome, some cosmetic or skin genomics? Do you think that these are some what people call pseudoscience or some people call preventive medicine? What is your opinion, sir?
Yeah. You look at the advance in the in the past 30 to 50 years. You see the great advance in genomics, and one of the concern that you raised in the slide is, do you think that in next 30 years from now, you still think that the genetics still exists as a specialty, or it's become more general and assimilate in every single specialty?
I think that's actually very relevant. When I started genetics, you could do the whole field from pediatrics to cancer, and I think that's now very much more difficult. And probably what we want to aim for is to make sure that our undergraduate and our postgraduate education gives everyone genetic skills. And so I think a lot of the genetic specialty will be teaching others the principles of genetics. But I can't see, looking 30 years ahead, that we will have a general geneticist who can cover all areas of genetics, both clinically and in laboratory medicine. So the way around that is probably for genetics to become more super specialized into different parts of the body and different disease systems with time.
And so now Thailand is starting to do the whole genome sequencing. How many sample that you think that it should be relevant for our whole genome project? 100, 1,000, 10,000 or million?
I think to get the background variation, the answer is the more samples, the better. I think one of the problems that you will have in Thailand and indeed in many other countries outside North America and Europe is you've got a different population, and therefore there's going to be a lot of genomic variation that's not at the present time as well recorded as perhaps it is in America and Europe. So I think you'll probably need to do a large number of samples from local populations in order to understand the variation in whole genome sequencing.
And another question is regarding to the genetic counselors. This is important. And how many genetic counselors in UK now, roughly?
There's certainly been an increase in the number of genetic counselors. I think, I don't know if I've got a figure, so it's certainly several hundred in the UK. It may be that with time that a lot of the counselling will be done by other specialists outside genetics. So there has been a move for primary care physicians to do some of the basic counselling. And I think if we look at the manpower, it's going to be very difficult to provide the number of genetic counsellors for all the genetic data that's coming through. So I think it's essential not just to train genetic counsellors, but to train other specialists to do some of the basic counselling as well.
So do you think that we will be able to finally understand the whole genome letter by letter throughout like 3 billion letters instead of like understanding only for the coding sequence at the moment?
Sorry, I didn't quite catch the question.
So do you think that we will finally one day be able to understand the whole genome sequences, the letter by letter?
The way that the knowledge in genetics is increasing, we are going to have an exponential increase in our understanding. I think there are going to be a lot of surprises that we can't anticipate at the moment. So it's not just a matter of knowing the genomic sequence. There's going to be some interesting discoveries about gene expression and perhaps how the spatial configuration between genes on the chromosomes will modify the expression of genes. All that sort of level of understanding has still to come. I think in my talk I suggested that if we looked at this in terms of child development, we're at the age of about 1 to 2 years of age when we're beginning to get the first words and understand a few words. But we can't put those words together into sentences, and we're certainly not writing in paragraphs, and we're not writing literature and poetry with the genetic knowledge at the moment. So a very long way to go, but a very exciting journey.
As anyone know that in Europe and UK now being hard hit by the SARS-CoV-2 or COVID-19, as you are well renowned, the geneticists, do you have any part to help or support that activity to reduce that impact at all?
I'm not actively involved with the COVID testing as I retired as Group Medical Director about 18 months ago, but my successor has been very much involved in developing some of the testing. And one of the things that the geneticists at the Francis Crick Institute are doing is sequencing the COVID virus to see if there's any mutations developing and if any of those mutations cause increased pathogenicity. And I think that's very important because even if we get a vaccine, as it looks as though we will, there may be further mutations that require new vaccines in the future. So I think that's the role that geneticists have to play at the moment in the COVID crisis.
Well, so do you come to an end, and thank you so much, Professor Patton.
Thank you very much for
Thank
the
you, sir.
invitation.